name is Manolis Kellis. I'll be giving a primer on the non-coding genome part of this series, and I'll be talking about how we can use genomics and epigenomics to understand the circuitry of human disease and to also start manipulating it. So, as you know, genetics, human genetics, has completely transformed our understanding of many diseases by being able to systematically map out across the genome in an unbiased way all of the genetic regions that are associated with a particular disease. We can learn about new genes that were previously unsuspected to be related to the disease. And that can open up across many different regions in the genome, many loci that are associated with disease. It can open up entire pathways that are repeatedly disrupted, but for which we didn't actually know that they were involved in the disease. And that can in turn enable discovery of new target genes, new therapeutics, and ultimately personalized medicine, whereby we can actually use the genome of a person in order to actually tailor the treatment to the specific mutations that they carry. So, how do we do that? We basically need to go now into these regions and look at what are the genes associated with these regions. And most of the time, what we actually do is that we simply annotate the region of association, which often spans multiple genes, with one of the genes that we hypothesize is the causal one, is the driver one. And when you look at therapeutic development, that's the gene that people are going to go after. That's the gene that people are going to be focusing on. And in this particular case, I'm showing you the genome-wide association study, Manhattan plot, for BMI, for body mass index, an index of obesity. And what you find is a lot of genes across the genome, but the strongest hit by far is sitting in this region that contains a gene called FTO, which was promptly renamed fat and obesity associated based on this association. And when I told you that in most cases you have many genes in that region, so you're uncertain what is the gene that's driving all this, in this particular case it was very clear this entire region of association was localizing very precisely to the first intron of this FTO gene. So people were like, great, let's focus on FTO. And they spent probably millions, if not billions of dollars trying to develop therapeutics, try to understand the biology of FTO. Turns out it's a methyltransferase, so basically said, oh great, RNA methylation must be super important in obesity. And you can still see reviews talking about RNA methylation being important in obesity, all based on the fact that FTO is an RNA methyltransferase. Turns out they were completely wrong. So FTO does not appear to be the causal mechanism underlying this, even though it's the only gene in that region. How is that possible? The reason why it's possible is because of all of the genetic variants that are associated with obesity in that particular region, none of them perturbs the protein sequence of FTO. They perturb non-coding nucleotides that are surrounding this particular exon here and this particular exon here. And that's not an exception. If you look across disease studies, you find that in 90% of cases, we don't actually hit the gene directly. We don't actually hit the protein coding region of the gene directly. What we hit is the non-coding region surrounding a gene. And very often people make the very natural assumption that, oh, it must be the gene nearby just because it's sitting right there. But more than half the time we'll be wrong. Why? Because when we don't have a coding mutation, it could just be a regulatory element that controls some other gene. Control elements in the human genome don't need to necessarily control the nearest gene. In fact, there's very strong examples now of very long-range regulation. And in this particular case, we actually found that the causal variant underlying this association controls two genes up to 1.2 million nucleotides away, jumping over several nearby genes, completely changing the underlying story for how FTO actually works in the genome. And that's what I'm going to tell you about today. So the challenges that we're going to be addressing in this primer is how do we start from a genetic region of association and look for the target gene? In this particular case, it's two different genes very, very far away. How do we find the causal variant? Namely, there are many nucleotides that are associated with every one of these genetic regions, but not all of them are causal. The vast majority of them are just simply passengers that are simply co-inherited, but are not the drivers of the phenotype. So how do we find these drivers? How do we find what cell type it even acts on? When a mutation targets a protein-coding gene directly, we can say, well, where's the gene expressed? And then you can maybe look at one of the tissues where the gene is highly expressed. For non-coding variants, where do you start? Do you start with where this gene is expressed or where all of the nearby genes are expressed? So what we need is maps of activity of non-coding regions that are independent of the expression of genes. And we're going to use epigenomic marks as a way to get at that non-coding activity. And then lastly, how do you go after the actual mechanism? What are the pathways that are altered by this association? What is the cellular and what is the organismal effect of these mutations? Everybody with me so far? So, how do we do all that? The beauty of being in 2016 and not in, 2001, is that we are now starting to have more and more complete maps of the non-coding genome. So when we all started out, people were like, hey, let's just sequence the genes of the human genome, and then that'll give us all the disease stuff. And then, somehow the argument was won after much, much effort that we should actually sequence the whole human genome, not just the protein coding parts. And then we basically said, let's annotate the genes. And there was a lot of effort to also annotate the non-coding regions. And that's where the field of epigenomics and comparative genomics basically opened up new doors for understanding what people have dubbed the dark matter of the genome, namely the 99% of the genome that does not code for genes. And there's been a lot of systematic efforts by the NIH and by other sister organizations to support the annotation of the non-coding genome. And all of this effort is now starting to pay off. And that's what the ENCODE project and the Roadmap Epigenomics project and the Blueprint project and the International Human Epigenome Consortium, or IHEC, have tried to do. They've tried to basically generate a systematic annotation of the non-coding genome across a bunch of different cell types. Because the human genome is more or less the same in most cell types with a few striking exceptions, but the human epigenome is dramatically different from one cell type to the other. And that's what gives the diversity of the human body functions. The fact that I can have neurons that act in completely different ways than glial cells that they co-inhabit, that act in completely different ways than heart and muscle and lung and bone and blood and so on and so forth. And I can have like 20 different types of immune cells, each one with their own program, even though they all have the same genome. And the reason is that the epigenome is dramatically different from cell type to cell type. So that's why the Human Epigenome Project had to go after a diversity of cell types rather than just a single reference. And that's what we have generated, and that's what we've published, and that's what I'm going to describe in the first part of the talk. Once you have this annotation of the non-coding genome, you need to actually link non-coding elements to their target genes. So if I have a mutation here, I'd like to be able to say that it affects this gene or that gene or that gene. And in the case of FTO, that it affects a gene that's like more than $1 million nucleotides away, $1 billion away. So how do we link these non-coding control elements to both their target genes and their upstream regulators in order to understand who is the controller that flips that switch normally and what happens when that switch is broken? You need to actually understand the controller which actually flips that switch. And how do we understand the intermediate molecular phenotypes and cellular phenotypes? So this is what, the promise is, the remedy to sort of the challenge that we have. So what we're going to deliver is, number one, for every region of genetic association, what is the relevant cell type? Number two, what are the target genes? Number three, what is the causal variant? Number four, who is the upstream regulator? Number five, what is the relevant pathway? And number six, what are the intermediate phenotypes? So these are the 6 things that we'd like to know for every one of these non-coding associations. And how many are there? There are thousands of those. So, 90% of GWAS hits require this. And now there are thousands upon thousands of genetic association studies, results that are simply sitting uninterpreted. So we have all the data there, we have all of the annotations, we have all of the genetic results. What we're missing is the interpretation, the mechanism. So what I'm describing today is a very general technique for doing that. So let's go through it. What we need to do is, number one, map and characterize the circuitry of the non-coding genome. Number two, identify what are the disease-relevant tissues, variants, and regulators, and we're going to do that by looking across multiple regions associated with the disease and looking for enrichments, for common patterns between them. Number three, we don't have only the ability to look at genetic variation between individuals. The epigenome varies as much as the genome, if not more, from person to person. So we're going to be combining genetic variation between individuals with epigenetic variation, with how my neuronal epigenome looks compared to your neuronal epigenome, not just our genetic makeup. And we can combine the two together to get at the disease mechanism. Number four, how do we put all these together to basically go after that first slide, the FTO locus, and uncover the circuitry and the mechanism of the disease, and use that circuitry that we have uncovered to now start manipulating the disease mechanism. To basically say, okay, well, if that's the circuit, I can intervene here, here, and there, and if I flip it this way, it will go one way, and if I flip it the other way, it should go the opposite way on the phenotypic level. And then lastly, how do we deal with the fact that there are hundreds of disease mutations that are scattered across the genome and that are very different from person to person? How do we deal with the heterogeneity of the disease and the very weak effects and the accumulation of these very weak effects? And this is particularly relevant when you start getting to the rarer end of the spectrum, where you basically have these humongous number of different hits that start emerging, each of which has very, very small effects. And this is particularly true in the case of cancer, where heterogeneity is, one of the leading paradigms of how we understand cancer. So the first part is basically how do we map the non-coding genome and its circuitry, and this is something that we published as part of the Roadmap Epigenomics project, which I'm going to be describing here, but also as part of ENCODE, IHEC, Blueprint. There's been a number of papers that have recently come out that paint a more and more complete map of the human epigenome. So I have to acknowledge that this is, an incredible collaboration with labs that are spanning both the US and outside the US, in particular John Stamm, Bing Ren, Alex Meisner, Joe Ecker, Brad Bernstein, Joe Costello, Martin Hurst, and on the computational side, Alex Milosavljevic, and this has truly been an incredible effort spanning many, many, many years and many, many labs. So what was that effort? That effort was to basically systematically gather more than 100 different human tissues and cell types and map the epigenome for each of them. So what are they? Number one, we have adult tissues and cells, brain, muscle, heart, digestive, skin, adipose, lung, blood. Number two, we have fetal tissues. And number three, we have embryonic stem cells, induced pluripotent cells, and differentiated cells that are derived from these cells in vitro differentiation. And for each of those, we basically go after numerous epigenomic marks. So what is the epigenome? The epigenome is basically all of the highlights, the annotations, the margin notes on the book of life. So you basically have, every cell has 23 volumes of an encyclopedia of all functions that will ever be needed to any of human cells. It's kind of daunting. It's kind of like, here's everything you'll ever need to know, regardless of what profession you'll end up doing. And if you end up being a physicist or a plumber or an artist, you'll need a different set of that knowledge. And that's sort of what the annotations on that book are. So basically, depending on your profession, you basically focus on different chapters. And for every one of those chapters, you focus on different paragraphs and different sentences. And you highlight them with, highlighter, or you underline them, or you put post-it notes. And that's exactly what the epigenome is. These are all the markings that allow every single cell to interpret the common book of life, the common genome that it has inherited, in its own profession, based on being a neuron, glial cell, oligodendrocyte, and so on and so forth. So how are these marks put on the book of life? So DNA is here, that we know and love, the double helix, the most noble molecule of our time, and it's wrapped around nucleosomes. And nucleosomes are basically bundles of 8 histone proteins that have long amino acid tails sticking out. And the post-it notes are effectively the fact that you can, put a marking on the histone level. So there's 147 nucleotides wrapped around that histone protein, and approximately a 50 nucleotide linker that can vary depending on how compact chromatin is. So at the 200 nucleotide level, you basically have, different colors of chromatin. And that's the first level of annotation. That's histone modifications. And we'll talk about those as histone H3, lysine 4, trimethylation, histone H3, lysine 4, monomethylation, histone H3, lysine 36, trimethylation, and so on and so forth. And I've color-coded those according to promoter, enhancer, transcribe, repressed, heterochromatic, active functions. So every one of those is associated with different functions of the underlying chromatin. Is everybody with me so far? Great. The second level is the accessibility of DNA to transcription factors, to regulators that like to bind in a sequence-specific way. And you can see that between nucleosomes, there's a lot more binding of regulators. And when they do bind, they displace the nucleosomes. They open up the chromatin. So that's the second level. So basically, is the DNA accessible? The third level is on the DNA strand itself, there are 4 bases, A, C, G, T, but one of those bases has 2 different forms. C can be methylated, especially when it's followed by a G. Of course, there's exceptions to everything. A can be methylated and other modifications can happen. But CpG is by far the most abundant modification of DNA directly, and it is typically associated with repression. So in a control region, if you methylate that control region, you usually turn it off. In genes, it's a little different. When a gene is actively transcribed, it's actually more methylated, so it's a little backwards. If you see methylation, it doesn't always mean off. Inside the gene body, it means on. But that's probably because you don't want transcription to restart in the middle of a transcribing body. So sort of you turn off initiation from there. But anyway, DNA methylation is a little more complicated. Actually, all of them are pretty complicated. But these are the 3 forms of epigenetic modifications. So what the Epigenome Roadmap Project did is map these forms systematically by carrying out chromatin immunoprecipitation experiments, where you basically build an antibody that recognizes that particular modification, and then you use that antibody to pull down all of the regions of the genome that are associated with that modification. You chop up the genome beforehand, you cross-link it, you basically pull down these regions, and then you do next-generation sequencing. So you basically sequence what comes out. And computationally, you map that back into the genome, and you basically are able to say, aha, in this particular segment, there was a lot of this. And in that particular segment, there was a lot of that. Everybody with me? For DNA methylation, it's different. You basically treat the DNA chemically, and that changes all of the bases that are not CpGs, and then you compare the reference with, that are not methylated CpGs, and you compare the reference with the bisulfite treated version, and that's what whole genome bisulfite means. And then you end up with a comparison, and all of the differences are basically the places where, you can infer was methylated or unmethylated. And then for DNA accessibility, you basically try to chop up the DNA, and you look at where did it get chopped up. So this is the traditional DNase assay. ATAC-seq is slightly different. You basically throw in transposase, and it sort of integrates in the places where it's accessible, and then you can sort of map all of the bases where you're integrated, and that gives you a map of accessibility. Who feels that they learned something so far? Raise your hands. Awesome. Great. Fantastic. All right. So that's the basic technology. This is where we applied. Let's see what we learned. Okay? So now let's look at these maps systematically across a region of the genome. So here are the genes, 1, 2, 3, 4, 5, 6, about 10 genes in this region. PAX5 is, in the middle, a good gene to focus on. And you can now see all of these different modifications mapped in IMR90 lung fibroblasts. Okay? So what do we see? We basically see that, transcription, RNA-seq, which is the output in many ways, is, a very poor predictor of what's going on here. There's like 2 little blips. But then when you look at the epigenomic landscape, it's so much richer. You basically see here that DNA accessibility is found in a bunch of places, correlating with usually promoter regions and to a lower degree enhancer regions. You can see here H3K27 trimethylation. It's a mark associated with polycomb repression. It is actually found on top of this PAX5 gene, which appears to be repressed in this particular region. Then you look at DNA methylation. Remember that repressive mark is actually lower here, where polycomb, which is a repressive mark, appears to be. So you basically learn that there's different types of repression. The genome is not just on or off. It can be off in many different ways. It can be off in heterochromatin. It can be off in polycomb. It can be off in DNA methylation. So usually 3 different, stop signs. You also see the high C interaction. So this is chromatin conformation capture, and it basically tells you at a very long distance what are the pairs of DNA segments that tend to be in proximity to each other in the 3D nucleus. And here you're sort of learning about this long-range structure of the genome. And then you see these promoter-associated marks that I've labeled in red, and these enhancer-associated marks that I've labeled in orange. And what you can see here is in this particular segment, which is this massive, massive enhancer, you basically see H3K4 monomethylation is much higher, H3K4 trimethylation is much lower. You see this difference between, one mark changing to another mark, and that is actually telling you something about what's going on underlying all this in the genome. So basically, all of that information is extremely rich, and you need to know a tremendous amount in order to sort of start interpreting these genome-wide tracks. And the problem is that we have these tracks for 100 cell types. So we can't possibly afford to look at, 1,000 different tracks simultaneously. So we need ways to interpret this. And we've developed computational ways to do that using a very simple process known as a hidden Markov model. So you basically take a Markov chain, which is a repeating process, basically, that has a key property called memorylessness. So it only remembers the current state that it's in, and once you know the current state that you're in, you forget everything about the past. And you basically walk along the genome, basically saying, what state am I in now? Oh, I'm in a transcribed state. What's the state am I in now? Oh, I'm transitioning from transcribed state to enhancer state, and so on and so forth. So what we can learn is the underlying state of the chromatin, the underlying state of the epigenome, the status that it's in, the chromatin state. And the word is a little bit, loaded. It basically means both the hidden states of a hidden Markov model, as well as the underlying state of the chromatin, the biological state of the chromatin. And we can basically build this hidden Markov model, specifically a multivariate hidden Markov model that basically looks at not just one variable, but multiple variables being emitted at the same time. And it infers the maximum likelihood hidden state of the genome, of the epigenome in that particular cell type that likely gave rise to what the output is. Okay? Who followed this part? Raise your hands. Okay, there's still some computationally inclined people. That's great. So, to make a long story short, we basically are able to summarize these many different epigenomic tracks into a single track per cell type that basically tells us what state that part of the epigenome was in. And here we can basically label it as enhancer, and we color code that as orange. Then, we can call these promoters, and we color code them as red, transcribed regions in green, repressed regions in gray, and you can look at a glance, a summary of all of that, which basically tells you about the fact that this region has more H3K4ME1 and less H3K4ME3, rather than dealing with a single mark at a time. It deals with multiple marks at a time. Okay? Everybody with me? So we can now take this single track from IMR90 lung fibroblast and bring it with its 100 closest friends, all of the other cell types. And IMR90 lung fibroblasts are at the top there. And you can see, again, the PAX5 gene nicely repressed in this particular cell type. And then you can basically see how it turns on and off in the different cell types. So you basically have a dynamic view of the epigenome in a single picture. And you can basically here see that, specifically in this particular cell type, PAX5 gets de-repressed. The promoter turns on, this big enhancer in its first instance turns on, and then the rest of the gene body appears to be transcribed. And you can see that simply by the chromatin state annotation. And you can see how it's repressed in most other cell types, and again, it turns on here. You can also see how promoter regions, which are in red, are fairly stably on, even when the gene does not appear to be transcribed. It's kind of cool. It basically tells you that promoters are basically very stable landmarks across different cell types. But instead, where the dynamics come from are the enhancers. You can see the enhancers are much more dynamic in sort of turning on and turning off in different cell types. Everybody sees this? The other thing that you can see here is how this region here appears to turn on exactly when this PAX5 gene turns on. Even though there's a lot of intervening genes in the middle. So if I were to link this region, I probably wouldn't say that it controls any of these nearby genes. I would say that instead, it appears to be controlling PAX5. So the dynamics, because of the correlated activity patterns between control elements and their target genes, tell us something about the linking, not just something about the state of a particular cell. Everybody with me? And this is one way that we can do linking. We can basically say, when enhancers turn on, what are the genes that turn on together, and then maybe link this enhancer with that gene and that enhancer with that gene, even though they're crossing over. They're not the nearest gene each time. And that's one approach. The other approach I sort of pointed out earlier, which was the links that you could see below the epigenome. And this is high-C. It's basically chromatin conformation capture, carbon copy, and a bunch of other stuff, and that's why it was labeled high-C, because it had a lot of C's. And basically what this tells us is the long-range linking between any genomic element and any other nearby genomic element. And it's a very cool assay, and it basically consists of taking your noodle soup, that's like the genome, basically very, very long spaghetti, and then gluing it together, chopping it up, and religating segments that were chopped up. And what that does is that it creates little loops between things that were together in 3 dimensions, even though they actually came from very different strands if you were to unravel them. So basically by chopping them up and sort of religating them in this way, we're able to say that, this region was ligated quite often with this other region, suggesting that there's a 3D loop from that linear genome that's folded in 3 dimensions. Everybody with me here? Great. All right, so that's the second technology. And then the third technology for actually linking control regions to their target genes is QTLs. So these are quantitative trait loci. They're usually EQTLs, expression quantitative trait loci. So what does that mean? Basically means that you're going to use gene expression level as your quantitative trait. So rather than just using height of a person, you use expression of this gene in that cell type. And then you ask, what are the genetic markers nearby that correlate with whether that gene is highly expressed or lowly expressed in, say, adipocytes? So if there's a genetic marker that is frequently found in individuals that have a higher expression of that gene, it basically says that there's a control region that targets that gene. So if I look across individuals, I basically find that the individuals that carry a T show higher expression than the individuals that carry a G at that location. And that basically means that a G to T change at that particular location controls the expression of that gene, and therefore that there's a regulatory element here that is linked to that gene there. Did anybody follow this part? Raise your hands. Okay, good. Perfect. So these are the 3 technologies for mapping interactions between control regions and their target genes. On one hand, the physical linking of the 3-dimensional structure. On the other hand, the activity, the functional correlation in the activity pattern across different cell types. And thirdly, the genetic evidence of EQTLs with SNPs, single nucleotide polymorphisms, that affect expression of nearby genes. Okay? So having all that together, we can start addressing the question of what are these control regions targeting? But then there's the question of who is targeting these control regions. So the circuitry upstream of these control elements. And that's where the fact that these enhancer regions are very dynamic comes in handy. Because I can take all of my enhancer regions that turn on in the same cell type and now say, across the entire genome, what are all of the enhancers that turn on in brain? Can I now look for common sequence patterns inside these enhancers? So we're going to now look at, all of the enhancers saying active in liver and say, what are the sequence motifs, the sequence patterns that are repeatedly found in all of those? And what we're finding is that hepatic nuclear factor 1b is indeed very strongly enriched in liver, and FOX4 is enriched in rectal mucosa, and RFX4 is enriched in brain, and that's exactly what the biology tells us. So basically, we're recovering known regulators, but in a completely unbiased way. We're basically saying that if there's a mutation that disrupts this particular motif, I'm more likely to associate it with function, and I'm also more likely to associate it with this particular upstream regulator. So we're sort of putting together the pieces for painting that circuitry. So having gone through all this, we can now go back to any genetic region of association and look at all of the SNPs that are associated with it. In this particular case, it's only 4 SNPs that are co-inherited in that genetic region. And I chose this picture because it's simpler, but in practice, there are dozens, if not hundreds, of SNPs that are co-inherited. And then we can ask for every one of those SNPs, what enhancers does it overlap? And what cell types are these enhancers active in? And who are the regulators that are predicted to control these enhancers based on these motif enrichments? And what are the target genes that are predicted to be controlled by changes in these enhancers based on the 3 lines of evidence that I mentioned earlier? So we can put all that together and come up with a much more rich picture. So instead of just basically saying, hey, there's a region here that is associated with disease, we can have very specific hypotheses for every one of those SNPs. What is the target gene? And you can see that if you predict that this SNP is causal, you predict that this is the target gene. If you instead predict that this SNP is causal, you predict that the function is instead driven by these 2 other genes. So understanding the specific variant means understanding the specific regulators, means understanding the specific target genes, understanding the specific cell types, and so on and so forth. And all of that information is interlinked, and we have developed methods that allow us to sort of iterate through and try to sort of find the right combinations that increase the overall probability of understanding the mechanism of that region. So that's where we're at. We're basically able to now systematically start making predictions based on this massive experimental data and a lot of massive computation to interpret that data. We can start making predictions about every single region in the genome. And we can put out these predictions to the community, and that's exactly what we've done. We basically developed a program called HaploReg, for haplotype regulation, that's been used millions of times. And what that program says is basically you go on the web and you type the favorite SNP that you care about. And he basically says, well, hey, don't think at the level of SNPs, think at the level of haplotypes. So here are all of the SNPs, the single nucleotide polymorphisms that are co-inherited with the C2T mutation. There's a T2G mutation, C2G mutation, G insertion, and so on and so forth, that you should really consider because they are at almost perfect linkage disequilibrium with your SNP of interest. And the mechanism could be mediated by any of them. And you can now ask for every one of those, what are the overlaps with evolutionarily conserved elements? What are the overlaps with promoter histone marks, with enhancer histone marks? And not only do they overlap, but what tissues do they overlap in? This one is particularly active in adipose and, so on and so forth. What are the open chromatin regions that it overlaps with? What are the proteins that are bound there based on ChIP-seq, chromatin imprecipitation followed by sequencing? What are the regulatory motifs that are specifically altered by this mutation? So the C2T mutation is altering this particular motif, and what are the genes that are nearby? Okay? Everybody with me here? So this is sort of what we could have stopped at. We could have basically said, great, we're done. We basically did all these experiments, we mapped the non-coding genome, and then we sort of predicted these links, and then we can go in and make all that information available to the community, and we're done. But the problem is that there's a lot of data here, and it's very difficult to sort of get at the mechanism simply by looking at this, because there's a lot of hypotheses that you should be pursuing. So what we need to do instead is actually have a prioritization, not simply an annotation. This basically allows you to exhaustively mine, and also exhaustingly mine, all of these regions and basically say, here's 100 hypotheses that could be going on here. But what we'd like to know is, what are the most likely ones based on the genome-wide enrichments, okay? And that's the next part of the talk. If you guys are not exhausted, maybe you could stand up right now and actually take a stretch break. We're going to do a 30-second stretch break. So stand up, everybody, even everybody watching on YouTube. Stand up from your chairs right now and just stretch up because we need blood flowing to your brain cells for the next section. It's mandatory. Please stand up. Russians don't need to stand up. That's for the weaklings. All right, so part 2, let's now look for some enrichments. So we've done all this annotation, let's now look for enrichments. So what are we going to do? For every trait, we're going to look for all of the associated regions in the genome. So if I have height, for example, we now have probably 100 loci that are associated with human height, 180 in the last publication, where you basically have, different regions in the genome that contain within them individual SNPs that are very highly associated with height. And then if you look at type 1 diabetes or blood pressure or cholesterol, you actually get different regions of the genome, which, obviously makes sense. And that's where it gets kind of cool. Because you can basically take these regions and use them as controls for each other. You can basically say, what are the enrichments that I find in height versus cholesterol, versus blood pressure, versus type 1 diabetes. This way you're not just biasing for regions that have long haplotypes or common frequencies or you name it, but instead you're really looking at the biology of each disease. And then for each of those, we can do the following exercise. We can basically say, let's take all of the enhancers that are active in embryonic stem cells. And let's simply ask, which of those enhancers overlap genetic variants with each of those traits? And what you find is that, they much more frequently overlap height-associated regions than other regions. And you can do the same thing with immune enhancers and just, paint them on the genome and then simply ask, what traits do they overlap? And they overlap type 1 diabetes more than the other traits. So you can basically put another check mark there. And you can do the same for blood pressure. You basically see that they overlap heart enhancers more, often than not. And then you can look at all of the genetic variants associated with cholesterol and ask, what do they overlap? And they overlap enhancers that are active in liver. So what you end up with is this diagonal that basically tell you that the genetic variants associated with different traits are enriched in enhancers that are active in different cell types. So that basically means that if I want to understand the biology of type 1 diabetes, I should be focusing on immune enhancers. If I want to understand the biology of height, I should be focusing on embryonic stem cells, and so on and so forth. So basically, this is taking us a step further than what we had before. Before, we were basically saying, what's everything that overlaps? And that was a lot of really hard information to gather, but it was all put together in one spot. Now we're going a step further and saying, from all of these associations, you should probably be focusing on these guys because they are the ones that are statistically enriched. Is everybody with me? Great. And you can use different enrichment tests and so on and so forth. But once you have that, you can now start expanding this to hundreds of traits and hundreds of cell types. So this is a cartoon, obviously, but this is the actual data from, the next figure. So if I go now across 58 different traits and 127 different cell types, what do I find? I find that indeed, if I look at genetic variants that are associated with height, they're not enriched in any of these cell types except for embryonic stem cells. If I look at genetic variants associated with any of these immune traits, like type 1 diabetes, multiple sclerosis, and rheumatoid arthritis, you basically find that they're systematically enriched in immune cells, but not in these other cell types, for the vast majority of them. If you look at blood pressure, that I had on the previous slide, you basically see no enrichment whatsoever, except for one, and that's the left ventricle of the heart. And that's exactly where the blood pressure gets built up prior to being pumped in the rest of the body. So enhancer changes in the left ventricle of the heart are basically causing differences in blood pressure across individuals. It's kind of weird, right? It basically tells you about the cellular biology, it tells you about the relevant cell type, it tells you something about the trait, and it also tells you that it's a regulatory trait. We're not looking at coding here, we're really looking at the non-coding genome. There's something going on in the enhancers of the left ventricle of the heart. If you look at fasting glucose-related traits, that's basically type 2 diabetes, you basically see down here the strongest enrichment is found for pancreatic islets. And again, for those of you who know a lot about type 2 diabetes, you know that the pancreatic islets are basically the target of this immune response, which is actually killing them off, and that's sort of causing this insulin imbalance in the body, and that makes a lot of sense. You know, it took decades of biology to figure it out, and now looking at this map, we have a very clear picture, you know, or at least a very clear prediction simply by looking at this matrix. If you look at cholesterol, no matter how you measure it, you basically find this dramatic enrichment here in exactly one tissue, and that's the liver. And that makes a lot of sense. If you look at inflammatory bowel disease, let's play a game. What do you guys expect? Inflammatory, immune, right? Inflammation. Bowel disease, digestive, right? So that's good. Inflammatory, immune, sure enough, that's great. And digestive is right there. All of it lights up. This seems to work, right? Who wants to take a bet for Alzheimer's? Alzheimer's disease. Pick a tissue. Brain. CNS. We look at Alzheimer's disease, second to last line. We look at CNS. Oh, gosh. There's absolutely nothing there. Sorry. How much did you bet? We'll be collecting afterwards. Good. Bingo. Ding. Doubled your bet, I hope. We basically have monocytes, CD14 plus cells. So CD14 is a mark of both macrophages and microglia. What are microglia? They're basically the resident immune cells that actually live in your skull before your brain gets there. So they precede the neurons in the developmental path, and they actually help prune the neurons, they help feed the neurons, they help clean up the neurons, they help support the neurons. They're basically essential to the well-being of the neurons. And if you look at Alzheimer's disease, you basically have this amyloid beta accumulation that ultimately results in these tau, aggregates that ultimately kill off the neurons. And what this is telling us is that it's not so much about neurons. It's more about the glial cells, the microglial cells inside the brain. And therefore, it's actually starting to tell us that Alzheimer's might be an immune disorder rather than a neuronal disorder at its causal basis. And indeed, when we collaborated with Li Hui Tsai in her lab to basically look at the progression of Alzheimer's in a mouse model, we found that there were indeed two changes. There were immune activation and neuronal repression changes in the brains of these mice. But if you look at the temporal component, you found that the neuronal repression happened at a later point, whereas the immune activation happened at an earlier point. Again, indicating causality, whereby it starts in the immune dysregulation and it ultimately results in the neuronal loss. And if you look at the enrichments across all of these different tissues, you find that brain is actually the least enriched, whereas immune cells are the most enriched, indicating that inflammation and immune component might actually be the causal component of Alzheimer's disease, which completely changes the therapeutic program. You're now going to start focusing on the cause rather than the consequence. And sure, you can sort of go after tau and you can go after the neurons and try to make them healthier, but what's actually causing that is the amyloid beta and the immune dysregulation, the microglial, which again sort of leads to a completely different approach to understanding Alzheimer's. And I mentioned that we've developed these computational models for doing this systematically. So instead of just doing this one cell type at a time, we can basically start combining all that together. We can basically deal with multiple diseases simultaneously. We can look at enrichment across hundreds of annotations at once. We can basically use all that to start integrating the epigenomic annotations, the genome-wide association studies, summary statistics, the linkage disequilibrium in these haplotype blocks, all of this together to basically predict for every SNP in the genome a probability that that SNP is causal for that disease in that cell type. Okay? And we've done this and we've published this, and this is called Riviera, which stands for something really cool, like risk variant inference with epigenomic reference annotations. And when you apply Riviera to Crohn's disease, you basically see that it prioritizes, based on the genome-wide enrichment, these immune annotations. And then it goes back into individual regions that have a lot of different SNPs that are co-inherited and co-associated. And it basically says, well, these are the top SNPs based on my epigenomic annotations across the whole genome. So it gives you a posterior probability of causality for every one of these SNPs. And then if you look at schizophrenia across the entire brain, it basically prioritizes the central nervous system. And it basically says, well, in this region of association, these are probably the likely SNPs. that one should be focusing on. And when we ask, well, are these predictions making sense? You can basically look at the enrichment for conserved regions or the enrichment for expression QTLs and digital genome footprints, a high-resolution form of DNA hypersensitivity. And you can basically see that the SNPs that we have prioritized by combining the genetic and epigenetic annotation together are much more biologically enriched than the SNPs that we would get simply by the genome-wide association studies. Is everybody with me here? Great. So we can go a step further. We can basically say, well, great, we have a way to prioritize individual SNPs within genetic regions that are already genome-wide significant. But what if I did this for genetic regions that are not yet genome-wide significant? So there's a very stringent threshold at 5 times 10 to the minus 8 for genome-wide association studies. And there's many variants that lurk just below that threshold. And, you know, some of them are real, and with larger cohort sizes will go above that threshold. Other ones will actually disappear with larger cohort sizes because they were noise. But we can use the epigenomic annotations to actually try to, you know, maybe get a little bit ahead of ourselves in terms of predicting what will the next cohort size tell me about this particular region. So we tried this in the context of heart arrhythmia, and we basically went with these genome-wide significant loci that we used to train an epigenomic model that basically told us what are the annotations that are the most enriched, and then use these annotations to go back into this twilight zone below genome-wide significance, so between 10 to the minus 4 and 10 to the minus 8, where, you know, there's all these loci that are sort of begging to become significant, many of which are wrong, some of which are right, and we're able to prioritize individual SNPs that are sometimes at 10 to the minus 4 or 10 to the minus 5, which is 3 orders of magnitude below significance. They're 1,000 times less likely, simply based on the genetics. But based on the epigenetics and the genetics together, we're now predicting that they're causal, and we're able to now go in and validate those experimentally. We're able to now start predicting whether they will show enhancer activity and go and test that in a luciferase assay and show that indeed if we perturb the SNP, the luciferase activity will change. You can basically look at these high C type interactions, these chromatin information capture interactions with nearby genes, and sort of find that indeed these regions are predicting target genes. And you can now go to these target genes and say, are they enriched in other traits that are related to this particular QT interval trait? And indeed, there's a very strong enrichment, you know, compared to stringent control. And if you look at mouse phenotypes, you basically find that the genes that are predicted to be targets, when you knock them out in mouse, indeed lead to cardiac conduction or contractility phenotypes much more often than what you would predict by chance if you match the epigenomic data or if you match the GWAS alone. But combining the two together gives you this dramatic enrichment. And then for individual candidates, we went in and used morpholinears to knock down that gene in zebrafish and looked at the heartbeat and measured the time of that heartbeat. And we found that indeed the heartbeat rhythm changed based on deleting these genes that are sitting in these loci that are 1,000 times away from becoming significant. So this is a pretty big deal because it allows us to now sort of start prioritizing new targets well ahead of, you know, when the genetic information will eventually get there. So what have we talked about so far? We've basically mapped and characterized the circuitry of the encoding genome, and then we looked at enrichments to identify disease-relevant tissues, variants, and regulators. We basically built these Bayesian methods. I mentioned Riviera. We also have Converge, which basically looks at the whole regulatory network circuitry to predict causal SNPs, target genes, and transcription factors. And we found, in the case of Alzheimer's disease, that the causal cell type might be immune cells rather than neurons. And in heart disease, we're able to identify new loci. Now I want to take this to the next level and basically say not just look at genetic variation, but also look at epigenetic variation. So genome-wide association studies basically take a very big leap between a single nucleotide somewhere in your genome and a organism-level visible phenotype difference from person to person. And that's a very big gap. It basically says that out of 3.2 billion nucleotides, a single nucleotide has a visible effect. I mean, that's, you know, dramatic. And that's the reason why many people were saying GWAS will never work, Well, turns out they were wrong. GWAS actually works amazingly well, and how's now giving us these thousands of loci, but it's still a very big leap that we're trying to make. What if we were to bridge that by looking at intermediate phenotypes? So we could basically specifically focus on individual tissues, and then look at, regulatory changes within these tissues, look at gene expression changes within these tissues, look at endophenotypes measured at the cellular level or at the organ level before we get to disease. And that allows us to now start bridging that gap and looking at small paths at a time, which basically gives us much more statistical power, because these phenotypes are much less complex. The further to the left you go, the less complex the phenotypes are, the less polygenic, the more oligogenic they are, where there's a small number of SNPs, of single nucleotide polymorphisms, that affect a particular enhancer, a small number of enhancers that affect a particular gene, a small number of genes that affect a particular endophenotype, and a small number of endophenotypes that affect disease. Whereas with the original picture of SNP to disease, you have thousands because they're converging along different pathways. So doing this, we basically went to, and of course the challenge there is that you have these nice unidirectional arrows coming out of the genetics, but all of these other arrows are actually bidirectional, because the gene expression difference might actually be a cause of the disease rather than might be a consequence of the disease rather than a cause of the disease. And the environment might actually be coordinately changing both the gene expression patterns and the disease. For example, if I'm exposed to particular chemicals, or if I have a high educational attainment, it might actually change my predisposition to Alzheimer's and change both disease and gene expression. So that's why we have to be very cautious that these arrows are actually flowing in both directions. So in the context of all this, we basically teamed up with David Bennett and Phil de Jager, and we basically went after these very large cohorts that David has gathered over multiple decades now of recruiting people in a longitudinal study. So these people enroll, and they have agreed to donate their brain post-mortem, and they undergo cognitive evaluations over many, many years, and they end up with this cohort where we basically have a very rich phenotypic information, but also access to a traditionally inaccessible tissue, which is the brain. And we can now go and measure in the prefrontal, in the dorsal dorsolateral prefrontal cortex, we can actually basically measure the methylation pattern or the chromatin inverse mutation level of particular marks, or the gene accessibility level, or the gene expression level, actually all of the above, in specific regions of the brain. And we can now play the following game. We can basically say, number one, what are the genetic changes that correlate with epigenetic changes? These are the QTLs that we talked about earlier, only instead of expression QTLs, these are now methylation quantitative trait loci. You can look at this correlation between epigenetic changes and disease, understanding that this is a bidirectional arrow. And you can also combine the two together to basically look at how epigenetic variation might be mediating the effect of genetic variants on disease. Who's with me on this? Great. So let's look at the first arrow. What are the genetic differences that correlate with epigenetic differences, and therefore that cause epigenetic differences, because we know that this arrow from genetics is always unidirectional. And what we find is the following. We find about 50,000 different regions in the genome where there's a methylation mark that is very highly correlated with a genetic difference. So that basically means that differences in our epigenome are driven to a large extent by differences in our genome, which is, it kind of makes sense that basically if I have a genetic effect on a phenotype, that genetic effect is probably going to have some epigenetic effect that's visible at the activity of these individual regulatory regions. And here I'm showing this overlay of a Manhattan plot for every SNP with the nearby methylation probes. And you'll notice that this axis is very different from traditional genome-wide association studies. This is 10 to the minus 300, which is, kind of nice to get. And the genome-wide significance threshold after Bonferroni correction for 1 million different hypotheses is all the way down here at 10 to the minus 10 over there. And you can basically see all of these different associations at different parts of that spectrum. Even all the way down here, you basically have this very robust association, a non-corrected p-value of 10 to the minus 12 with, between genetic variation and epigenetic variation. And, here, somewhere in the middle, you basically have this dramatic effect where if I know the genotype, if it's GG, GA, or AA, I can basically predict the phenotype, namely the methylation level, with fairly high accuracy. Everybody clear with this picture? So we can now start using that information to start predicting methylation from sequence. So we can get at this inaccessible tissue simply from the genetics of a person. And that allows us to do something very, very cool. It basically allows us to now learn a function from G to M in 800 individuals from the ROS-MAP cohort. And then use that function to predict methylation, to impute methylation. So from genotype to imputed methylation. But I can do that for 74,000 individuals because I have the genotype for a lot more people. And now I can actually start correlating the predicted methylation level with disease. Just like I did previously between methylation and disease, I could basically look at, methylation changes that correlate with disease. Now I'm looking at predicted methylation changes. And this is very cool in many ways. Number one, because I don't have to actually go and gather 74,000 brains. I can do that with a few hundred brains. Number two, I actually have specifically the genetic component of disease rather than the environmental component, which I have here. So this arrow is bidirectional, but this arrow is unidirectional. Who thinks that this is kind of cool? Awesome, great. So this is extremely powerful. You can basically now learn this function, bridge that very large gap by having an intermediate phenotype, and predict that intermediate phenotype rather than directly observe it, giving you the causality back. And here's an example. We can basically take a particular chromosome that only has one genome-wide association hit and suddenly combine multiple nearby SNPs to predict the methylation level of a probe, a methylation probe in that region, and then associate that probe with Alzheimer's disease. And what I get, using only genotype for these individuals, but predicting the methylation, is a lot more hits. So basically, I'm able to now start predicting regions where multiple genetic variants together are contributing to a change, a predicted change in methylation, which is then correlated with the disease phenotype. Okay? Yeah?
Does that bring you to take those that don't reach your threshold p-value?
Yeah. It absolutely. It's now allowing us to discover new loci that were previously not genome-wide significant. And the only reason we're able to do that is because we're using multiple SNPs at a time. Because if I only had a single SNP, then it's a linear function, and I would have basically found the association directly with the genotype. But because I'm using multiple SNPs guided by the methylation QTL information, I'm able to now surpass the limitations of looking for combinatorics in traditional genetic studies, because the combinatorics kill my statistical power because there's simply too many combinations, too many tests to correct for. Whereas here I'm actually guiding it from the specific SNP to methylation probe links that I predicted earlier. So we can do that at the level of methylation. We can do that at the level of methylation and transcription, and basically start predicting both, the alpha here between G and M, the gamma here between G and T, the beta between M and T, and ultimately disease for each of those. And then the other advantage of this is that it also tells us how the disease is mediated. Whether it's a particular methylation probe, it tells us something about that regulatory region. Whether it's a particular gene expression pattern, tells us that this might be the target gene through which it acts. And this is the relevant tissue where I'm predicting the methylation, where I basically built my methylation QTL. And we can also do that by adding surrogate tissues. We can basically say, well, methylation in the brain is inaccessible, but people are more willing to give a little bit of their blood. So why don't we use blood and genotype together to predict the methylation level in the brain? So I can measure expression in the blood, I can measure methylation in the blood, and somehow get additional information above the genotype that somehow captures the epigenetic differences or the transacting differences in regulatory variation between regulators for those people. And together predict that. And we're able to do this by predicting, across different tissues, one epigenomic mark to the other across different tissues, or by combining both the genetic and the epigenetic information together within these regions. And ultimately, that allows me to now start, yeah, please. Correct.
And here you are using set and the genome stays stable in all the tissues.
Correct.
How can we just take the epigenetic mark like DNA methylation from blood and...
Yeah, it's a great question. So I'm going to give you a toy example just to build your intuition. So suppose that in addition to the genetic variants that are acting in a particular tissue, I have some regulatory differences in a regulator. So basically, my particular protein coding sequence for this particular regulator is altered. And when that protein coding sequence is altered, it's altered in all my cell types. So if that regulator binds both in blood and in brain and has a transacting effect in both blood and brain, something that I will not be able to capture simply based on the genetic association in that region, by looking at these global changes that are happening in the blood for that person, I'm able to gain more information than if I have genetics alone for that person. Does that sort of answer your question? Yeah. So, I mean, it's a statistical test. The specific mechanism, we're sort of agnostic as to what it could be. It could also be that you have some environmental exposure that affects both your blood and your brain in similar ways. And somehow your epigenome in your blood is indicative of that exposure that now tells you something about the changes in the brain. So we're building a statistical model. If that model finds a correlation between blood and brain, the mechanism through which that manifests is going to be a challenge to dissect, but at least on the statistical level, we can already increase the power. I hope that answers your question. And we can, of course, start now using this to classify the pathways through which disease is acting. So if we now take multiple phenotypes together for a particular individual, like neuritic plaques, neurofibrillary tangles, neuroinflammation, cognitive decline, and so on and so forth, we can actually start measuring each of those phenotypes. We can start measuring covariates like age, sex, educational attainment, and so on and so forth. And we can ask which methylation probes across the genome correlate with which combination of phenotypes. And we can cluster the different probes across the genome together into effectively coherently associated subsets and ask, is there some common biology to these subsets? And the answer is yes. Basically, if you take the subsets that are associated with neuritic plaques and age, or neuritic plaques alone, or neurofibrillary tangles alone, or negatively associated with cognitive decline, namely, cognitive resistance, and so on and so forth, you can basically see that they're enriched in enhancers that are active in different cell types. So here are my 9 clusters here, and you can see this particular cluster lights up in embryonic stem cells. These other 2 clusters light up in immune cells. These other clusters light up in fetal and adult brain separately, in heart, and so on and so forth. So different parts of my epigenome are associated with different subsets of phenotypes and with different cell types in which they act. So even though they all converge to the manifestation of Alzheimer's disease, they're associated with different subsets of the biology of Alzheimer's. And if you look at the associated gene ontology terms for each of these clusters, again, you see enrichment with different pathways and processes. And I can do that with 3 phenotypes, or I can do that in the context of a whole clinical record. I can basically go in the electronic health record of an individual and basically start building metaphenotypes associated with these combinations. So I can basically, use neural net effectively to learn higher layer phenotypes from lower layer phenotypes. And this is very, very important in many ways. Number one, we can basically clean up these phenotypes. We can basically say that if I now take the medical record and split it into a number of metaphenotypes, I can basically start predicting these individual phenotypes going backwards. And I can do that with 80% accuracy in some cases, which is very, very encouraging. I can also start grouping individuals together based on these metaphenotypes. I can predict risks and order new tests based on what I predict will come positive, even though they haven't actually taken that test. I can look at a genotype association using larger cohorts because I only have these phenotypes in individuals that are a subset of the whole medical record. But when I can impute this, I can actually build much, much larger cohorts. I can perhaps identify more biologically relevant metaphenotypes, and I can also harmonize and correct these data sets. So this is a very active area of investigation, and we have several collaborations in that direction. So what I've told you so far is, number one, how do we map that reference circuitry? Number two, how do we identify the relevant tissues, variants, and regulators? Number three, how do we combine genetic and epigenetic variation to basically understand the role of these intermediate phenotypes? And now I'd like to go back to that very first slide and say, now that we're armed with all this, we're armed with QTLs, we're armed with these circuits, we're armed with these enriched tissues, and so on and so forth, can we now make sense of these regions? And let me remind you, what are the 6 things that we would like to know for every one of these regions? We'd like to know from a genetic region of association, number 1, what is the tissue and cell type that it acts in? Number 2, what are the target genes that mediate this phenotype? Number 3, what are the causal nucleotides among many nucleotides that are co-inherited in that region? Number 4, who are the upstream regulators that bind these nucleotides, whose binding is disrupted by the genetic variation? Number 5, what are the cellular phenotypes? And number 6, what are the organismal phenotypes that result from these target genes being altered? Everybody with me? Let's go through it for the FTO locus. Okay? So this is a paper that we published in collaboration with Melina Klausnitzer in the New England Journal of Medicine last year. And the goal is to basically understand the circuitry of these FTO obesity variants. So obesity is extremely important, as you know. In the US alone, it affects actually across the world, it affects 500 million people. In the US alone, it costs $200 billion a year. And the strongest association is what I showed you in the first slide, falls in the first intron of this FTO gene, and it contains 89 common variants, all of which are very significantly associated, any of which could be the causal one, none of which is protein altering. So given that there's no protein altering variants, people have hypothesized a regulatory role. But there's been a lot of debate about what is the target gene and what is the tissue in which it acts. There have been predictions that are very highly conflicting with each other. And very conflicting genes, very conflicting tissues. And there has been a focus on brain and appetite. And the idea is that, of course, obesity is associated with what you eat and how much you exercise. And therefore people said, okay, well, it must control the brain somehow. And that's what sort of causes you to exercise less or to eat more, and so on and so forth. But we basically said, well, let's go in a completely unbiased way and look at what are the tissues that light up across our maps. So here's the genetic region of association in the FTO locus. And you can basically see across the different tissues, there's this massive enhancer region. This enhancer region spans 12,000 nucleotides. I mean, think of it as a major control switchboard in the building. It's basically one of those master switches. And it's active not in the brain, as people had hypothesized, but instead in the mesenchymal stem cells that give rise to white and beige adipocytes. So you all know that we have different types of fat cells. We have good fat cells and we have bad fat cells. The good fat cells are the ones that burn energy. They're the brown cells. And they basically take calories and then they just burn them as heat by depolarizing the mitochondrial membrane and then causing a proton transfer and loss of energy as heat rather than storage of energy as ATP. The bad adipocytes are basically the white adipocytes that are sort of storing that energy rather than dissipating it. They store it as fat. These very rich, lipid-rich molecules, energy-rich lipid molecules that are basically sort of accumulating inside these white fat cells that become very large and they are full of lipids. Of course, I'm talking them as good and bad in, McDonald's age, basically. But in Ice Age, they were flipped, right? Being able to store a lot of energy was awesome because you'd go through periods of starvation. You'd have all that energy left over. Being able to burn all that energy right away was actually a very bad idea if you're going to go through a period of starvation afterwards. So the environment has now made this process be, the opposite of what it was in traditional sort of periods of less abundant calories and more activity in individuals. So this master switchboard is actually acting exactly in the white adipocyte progenitor cells that decide between these two phenotypes. And not between white and brown, but between white and beige. So beige is sharing the same developmental path as white, but it shares the same phenotype as brown, whereas brown is more related to muscle than to fat. Okay? So this is basically a switch that controls between, in the progenitors between those. So we basically said, well, maybe it actually decides whether you're going to become white or brown, or beige, but we still had to show that. So that was number one. Where does it act? So our hypothesis right now is adipocytes, and specifically pre-adipocytes, not brain. The second one is, what are the target genes? And this is where I showed you that the genetic association is very highly localized to this FTO gene. And FTO was actually originally named FATO for like a ridiculously weird reason, which is that the gene itself is very, very large. So they basically named this gene FATO, having no idea what it was going to end up being, and it was promptly renamed to fat and obesity associated, which is much more, palatable. But FTO basically, this very, very large gene, is exactly sitting on the genotype. But then there's all these other genes nearby that might actually be the target. So we basically said, let's look at high C data. Let's look at these high-dimensional chromatin conformation data and see what it tells us. And what it tells us is that this FTO locus forms these massive interactions spanning 2 million nucleotides and encompassing 6 genes on one side and 2 genes on the other side in these 2 very, very big domains. So any of these genes could be targets. They're co-localized together, so we have a mechanism through which they might actually be acting on these targets. But we don't know yet that the genetic difference affects the expression of these genes. To show that, we basically had to look at the relevant tissue in risk and non-risk individuals. So we basically went and gathered white fat, specifically pre-adipocytes in these white fat stores, from 20 homozygous risk individuals and 18 homozygous non-risk individuals. So having the risk haplotype or the non-risk haplotype. And then we basically asked, from all of these genes nearby, do we find any expression changes in adipose? And the answer was remarkable. No expression change for most of these genes except 2. IRX3, 500,000 nucleotides away. IRX5, 1.2 million nucleotides away. So these genes are basically sitting within this domain, here and here, but they're nowhere near the genetic region of association. I was at a conference a few weeks ago, and somebody basically said, oh, Manolis, who published the most depressing paper of last year. And the reason that they found that paper depressing is that they now have to question everything about every one of these regions where we basically call it the FTO locus, when in fact FTO might not be involved at all. I mean, I think that it's actually better to know that you're in trouble than to be in trouble and not know, so I wouldn't call the paper depressing directly, but, more, a warning sign. So anyway, we basically said, aha, it's one of those 2 target genes, and they're sitting very, very far away, and they're acting in adipose. So we basically said there is a genetic effect. The risk individuals have higher expression of these genes. And that's something to note, the fact that most of the time we think of disease as something is broken. In this particular case, something is overactive, right? So it's a gain of function. There's an increased expression for the risk individuals. And that's something that can be very easily done in non-coding mutations, whereas for coding mutations, most of the time you deactivate the gene rather than make it hyperactive. In this particular case, for non-coding mutations, you could simply delete a repressive motif, making that gene hyperactive. So that's number 2, finding the target genes. Number 3 is, what is the causal nucleotide and who is the upstream regulator? And I'm going to make a long story short by basically saying that using evolutionary conservation of motif modules, of combinations of multiple motifs, we basically predicted that there's one nucleotide here that is very deeply conserved across other mammals, but is changed from a T to a C in the risk individuals, thereby disrupting this AT-rich motif, and specifically this AT-rich interacting domain, ARID5B, for this protein ARID5B, which is actually very highly expressed in pre-adipocytes. So we basically have a mechanism. Out of these 89 common variants, we are predicting that this nucleotide mutation, this T2C alteration in the risk individuals, is causing ARID5B to stop binding, stop repressing the enhancer activity, resulting in this overexpression of RX3 and RX5. Okay? And we did a gazillion experiments to show this at every step of the way, and they're all in the supplement. But that's number 3 and 4, the causal nucleotide and the upstream regulator. What about number 5? What do these genes actually do? We basically said, well, great, RX3, RX5. I haven't told you anything about obesity yet. How do they act? So we basically said, let's look at the expression level of RX3 and RX5 across individuals. And look at what are the functions that correlate. We can basically look at gene ontology terms, groups of genes that are correlated with the expression of RX3 and the expression of RX5 across individuals. And what we found is that there's a positive correlation with lipid metabolism and a negative correlation with mitochondrial function. So when RX3 and RX5 go up, mitochondrial function goes down and lipid metabolism goes up. So that suggests a shift from energy dissipation to energy storage. And to validate that, we basically went to the cellular phenotypes and we said, well, do the risk individuals show less mitochondrial DNA content? And the answer is dramatically significant. They basically show, several-fold less mitochondria, suggesting indeed that there are fewer mitochondria and there's therefore less energy dissipation. And indeed, the adipocyte size becomes larger for these individuals. So they basically are storing energy rather than burning it. So this is the complete circuitry now. We basically have ARID5B, the upstream regulator, that normally binds this AT-rich motif and represses RX3 and RX5. In the risk individuals, you basically have this shift from a T to a C, disrupting this AT-rich motif, leading to lack of binding and overexpression of RX3 and RX5. This in turn leads to a shift from energy dissipation in thermogenesis to energy storage in white adipocytes. Is everybody clear with me? Great. So that basically allows us to now go from, oh, it's somewhere in the FTO region, must be the FTO gene, to, oh, there's a bunch of knobs that we can actually start systematically manipulating. I can basically go and overexpress or knock down RX3 and RX5 and shift between lean and obese. I can overexpress or knock down RE5B and shift between lean and obese. I can actually go and make a single nucleotide change in either risk or non-risk individuals and shift between lean and obese. So let's see if these actually work. We are now having knobs that we can manipulate. Do they work? The answer is surprisingly well. If you actually change a single nucleotide out of 3.2 billion nucleotides in the human genome, I mean, that's precision medicine right there. We basically can find that thermogenesis, this oxygen consumption rate, uncoupled as percent of basal, in presence of isoproternol, which is the brain agonist, is impaired in the risk individuals. Whether, the baseline oxygen consumption rate is very low, so they don't consume oxygen, they don't burn energy, and isoproternol stimulation has no significant effect, no change. Whereas if we take now the same risk individuals and we change that single nucleotide into a T, and we effectively rescue that disrupted AT-rich motif, we are basically able to restore the basal level of oxygen consumption rate. And when you stimulate it with isoproternol, you basically see a 7-fold increase in the level of the cellular phenotype. So that's remarkable, the fact that we can pinpoint that entire association to a single nucleotide, and changing that single nucleotide, fixing it, effectively restores thermogenesis in the cells of these individuals. I mean, that's remarkable. Then we can basically say, well, what about the organismal phenotypes? Great, this has an effect at the cellular level. What about at the organismal level? So we can go in and change the expression of the downstream target gene, IRX3, by basically building a dominant negative construct that's expressed in the AP2 promoter, which is active in adipose tissue. And what we can see is that the mice actually lose 50% of their body weight simply by having this dominant negative form of IRX3. And even more remarkable, when you put them, they start out leaner, but when you put them on a high-fat diet, the normal mice basically gain weight, The IRX3 dominant negative mice simply have no change in weight. You can basically feed them all the fat you want. They cannot gain weight. They're simply burning it off. So they're not changing their exercise. They're not changing their appetite. They're not changing how much they eat or how much they, go running. What they do is that they change their level of energy dissipation in this energy expenditure per body weight, both when they're active, when they're awake, and when they're sleeping. So they're simply burning off energy in their sleep, which is kind of nice, right? So this is basically where we're at. We're able to use all of that information to start predicting individual loci, and after a lot of laborious efforts, come up to the mechanism of these individual loci. But we'd like to do this a lot faster and at a much higher throughput. So we need to start dealing with heterogeneity. And heterogeneity is coming in many forms. Number one, for a particular individual target gene, there can be many changes in different enhancer regions. For a given enhancer, there can be multiple changes, either activating or repressing for that one. And in a particular pathway, you can have many changes in nearby genes, all of which are affecting that pathway. And you can have combinations of common and rare variants acting at both strong and weak effects. So this dramatic heterogeneity is something that we have to just embrace. And the way to do that is to actually build regulatory networks, regulatory models that allow us to, in a very natural way, incorporate rare and common variants, strong and weak effects, multiple enhancer elements, multiple mutations within an enhancer, multiple genes in a pathway, in a single sort of model, which is what we call convergence. So basically, from the motif to the enhancer to the gene to the pathway, we can basically look at how these mutations are coming together in a hierarchy of models. We apply this at the extreme level of cancer, where you basically have every patient having a different mutation almost. And what you end up with is this building of a regulatory plexus, this circuitry surrounding any one gene, where all of these elements are converging together. And we can now measure all of these mutations that are different across different patients happening in the same enhancer, and all of these enhancers acting at the same gene. And when we do that in prostate cancer, we find that there are multiple genes that become significant, with plexi that are either very dense or very sparse. But what's really interesting is that they're all converging in a small number of pathways. So yet there's another level of convergence at the level of the pathway, where you basically have immune, signaling, and mitochondrial functions that are repeatedly disrupted in these regulatory ways in the neighborhoods. So we went after one of them. We basically validated this PLCB4 plexus, where you basically have one gene here that's targeted by all of these control regions. For every one of them, we basically did a high C-type experiment with the gene itself, and then the region that we want to test, and then upstream and downstream elements. And what we found is that the region specifically interacted using 3C tests in every single one of these cases. These are the annotations of these regions. And this gene is in fact falling in the same pathway as PTEN, one of these hallmarks of prostate cancer. And what's really interesting is that it actually acts in the same synergistic level. So one could have predicted it simply by looking at the network diagram here, but the non-coding mutations would have been very difficult to elucidate without this, higher level convergence. And indeed, when we go and overexpress PLCB4 in PC3 prostate cancer, we find that it reduces activity of ERK and AKT, which is synergistic with PTEN. So we can validate the gene at that level. So we can scale that up to basically look at mutations systematically across the whole genome, whether they're common variants, rare variants, somatic variants, and start predicting their effect using existing data sets. And we can also start experimentally testing these regions using these massively parallel reporter assays. So we can basically test 10,000 or 100,000 of these constructs at once. You have seen several such recent papers. We published one of them, which is basically looking at this tiling approach for finding exactly where is the regulatory activity and where it's activating or repressive, being able to pinpoint individual regulatory motifs. And Vijay Sankaran has usually used this, as well as Pardisa Bedi, in order to go and disrupt individual SNPs. These are common variants. We're now using this for both common and rare variants within these regions. And we're also collaborating with Kevin Egan and his lab to basically start differentiating stem cells into multiple lineages to basically see which of these lineages actually show expression differences, epigenomic differences in a systematic way to basically ask what are the cell types where these common variants are acting. So that allows us to now do this in a systematic way rather than having to do this for every gene that we predict as a target. We can simply do this for all genes simultaneously by using a panel of heterozygous individuals and looking for allelic differences in those. And again, in the same grant with Kevin Egan, we're now looking into cellular phenotypes. So Kevin's lab is able to differentiate embryonic stem cells and iPS cells into mature neurons. You can see these neurons beating inside the 384 well experiment. And he's able to now start perturbing those and measuring calcium signaling and other cellular phenotypes in 384 well plates. So we can now start screening a very large number of these mutations guided by our computational models and then validating high throughput before going to the low throughput approach. And then lastly, in collaboration with Ron Weiss and his lab, we're now constructing circuits that are able to sense which sets of activators and repressors are present in the cell type of interest in order to be able to deliver a therapeutic intervention in that cell type. So if for IRX3 and IRX5, we basically predict that we need to act in pre-adipocytes, then we can basically build a signature for the specific set of microRNAs that are on or off in these pre-adipocytes, and then use them to make sure that our construct only turns on in those cells. So we package that construct in a virus, we infect the mouse, and then we basically look at which of those signatures basically allow it to turn on the right cell type, and that's where we can actually do a very specific perturbation to either overexpress or knock down that gene. And Ron basically showed that in the case of cancer, basically doing these very precise immunomodulation using these viruses, he's able to actually kill cancer in a very directed way when, with an empty vector, the cancer simply progresses. And that's by being able to sense the cancer state, rewire the cancer cells, and then recruit the immune system, and this has been very successful in vivo. So that's what I'm going to leave you with. Basically, we can systematically characterize the circuitry of non-coding elements. We can identify disease-relevant tissues, variants, and regulators based on these global enrichments. That was very surprising in the case of Alzheimer's. In the case of heart disease, it predicted new loci. We can combine genetic and epigenetic variation to basically identify what are the intermediate phenotypes that change. We can uncover and manipulate the disease mechanism and circuitry in individual loci. And we can actually start doing this systematically in a very high throughput fashion to basically perturb and start delivering therapeutically some of those interventions. So this calls for a completely new model of interaction between the pharmaceutical industry, academia, government, and basically, transitioning from this resource building and discovery, which has been traditionally the domain of NIH, to this commercialization and therapeutic development that has traditionally been the domain of industry, to basically an intermediate level of a loop between discovery, manipulation, and therapeutic development. And that basically calls for really a partnership between industry and academia rather than simply, us staying there and industry staying there. So we actually have a lot of partnerships that we're now starting with local pharma, which allows us to now start bridging this middle layer, which I think we're both lacking tremendously. So this would not be possible without amazing collaborators. So in the context of the Roadmap project, we basically had, it was led by Anshul Pundaji, Wouter Milliman, and Jason Ernst in my lab, who are now at Stanford, in Seattle, and at UCLA, each with their independent labs. Xinchen Wang did all of the subthreshold work in cancer in collaboration with Laurie Boyer. Matt Eaton led the Alzheimer's intermediate phenotypes in collaboration with Hilde Jaeger and David Bennett. Andreas Penning, who now has his own lab at CMU, collaborated with Liz Zoneska from Li Hui Tsai's lab for the Alzheimer's immune study. Melina Klasninger now has her own lab at Harvard, and we're collaborating very actively in several of these aspects. She led the FTO study. And then Richard Solari and Ron Weiss were our collaborators for the cancer circuit. Richard has now taken off in industry, actually in a new startup that tries to change cancer. And for the high throughput assays, it's led by Xinchen Wang, Sarah Goggin, in collaboration with Kevin Egan. So I'll stop there. and I'm happy to sit around and chat longer. Thank you, guys.